Hello everyone. Today, cell therapy are continuously improving and providing new options to cancer patients. In this talk, I will present you the optimization of long-term expansion of T cell inside the Eppendorf dicebox system, and more particularly, the impact of dissolved oxygen of this culture. A new type of cancer immunotherapy, also called adoptive cell therapy, ACT, hold the promises of a truly personal treatment. ACT is a form of living treatment as it is used the cells of our immune system to suppress cancer. Some of those approaches involve directly the isolation of our own immune cell and simply expand them. Other approaches involve genetically engineering of our own immune cell through gene therapy to leverage their cancer-fighting capability. Some types of ACT are now being used in the clinic to treat blood cancer like leukemia. T cells are one of the important types of white blood cell of the immune system and play a central role in the adaptive immune response. Those cells can be distinguished from other lymphocytes by the presence of a T cell receptor on their cell surface. Those cells are grouped in three subsets based on their function. We have first the cytotoxic T cell, also called CD8 plus cell, due to the presence of a CD8 receptor on their membrane. Those cells kill cells infected by virus, bacteria, but also destroy tumor cells. We have then the helper T cell, called also CD4 plus, due to the presence of the CD4 receptor on their membrane. Those helper T cells does not kill the cell directly. They send signal to inform the immune system how to coordinate an attack. Then we have the regulatory T cell, also called suppressor cell, who play an essential role in our immune system. Those cells reduce the activity of the other T cell when it's necessary. They can prevent T cells from attacking your body healthy cell. The development of a cell-based therapy product generally requires the production of a large quantity of high-quality viable T cells in a controlled environment. To generate larger cell amount, scaling out of the 2D approach was first initiated by simply multiplying cell culture vessels or by using multi-layer flasks as called cell factory or cell stack. Even with some automation for those 2D culture, this approach remains relatively cost, space, and labor intensive. In addition, those approach does not include any online monitoring and control of the key parameter of the culture, like cell count, dissolved oxygen, pH, glucose, and growth factor concentration. Therefore, the stirred tank bioreactor can offer a suitable environment for the culture of T cell by providing an homogeneous distribution of nutrient gas in order to have high process control capability. Therefore, the use of bioreactor system facilitates the development of a dynamic suspension culture and overcome environmental inhomogeneity typical of static condition. It becomes quite clear that the expansion of cell for cell gene therapy product require bioreactor as they allow process scaling up with high standardization and reproducibility. Those systems are equipped with technology for monitoring and feedback-based control of the culture environment, including basic and more specific parameters such as temperature, pH, concentration of nutrient like glucose, metabolite like lactate ammonia, and gases like O2, CO2. Therefore, the bioreactor became an essential tool for the development of efficient and high-quality cell therapy products. As you could see on this slide, the history of bioprocessing at Eppendorf followed closely the development of stem cell field. Eppendorf is a leading life science company that develops and sells systems for use in laboratory worldwide. The company Eppendorf was created after the Second World War with the goal to improve the people's living condition. After 75 years, the mission of the company remained the same. In between, in 2007, New Brunswick Scientific joined Eppendorf in order to enrich our portfolio with a broad spectrum of equipment for cell culture. In 2012, Eppendorf acquired Daskip Group. The high-quality bioreactor and software solution provided by Daskip nicely complement the current portfolio of New Brunswick. In parallel, in 2006, Yamanaka team of Kyoto University in Japan revealed a way of making embryonic-like cell from adult cell and avoid the need to destroy an embryo. This team were able to show to reprogram ordinary adult cell by inserting 4 key genes forming the induced pluripotent stem cell. In 2009, the first clinical trial with embryonic stem cell was approved by FDA. It showed the importance of closed, robust, and reproducible system. Now, in 2024, we are really focusing on cell gene therapy. The Eppendorf bioprocess solution includes 4 pillars: a scalable system, a single-use solution, software, and service and support. To achieve successfully therapeutic response, the development of efficient and high-quality cell therapy product has become a challenge for clinical researcher and healthcare provider. As you could see on this slide, Eppendorf bioprocess system cover a wide range of working volume from the small volume of 65 ml to 1,000 liters. Optimized protocol for the scalable manufacturing of T-cell are crucial, since T-cell and other lymphocytes require meticulous culture conditions. They are highly sensitive to their culture environment and could react easily by modifying their receptor ligand repertoire, which can cause change in cellular response to external substance. In this study, we have shown that parallel bioreactor control systems are useful to optimize culture conditions before the scaling up process. They provide a solid basis for optimal yield in subsequent steps from benchtop to production scale. Several studies have demonstrated that the culture of T-cells at different oxygen tension can have a significant impact on cell proliferation, redox status, and apoptosis activation. So, any small change in the culture environment can have a profound consequence for the product quality. In this study, we evaluated the impact of 2 different oxygen tension, 70% and 20% during the T-cell culture. Initially, the CD4 plus T-cell were expanded in a 2D static system in T75 flask for 4 days to ensure appropriate cell number for the culture in bioreactor. The seeding density was 1 to 3, 10 to 7 cells per vessel, and it was incubated at 2 different DO concentrations, 20 and 70%. The culture was maintained in bioreactor for 16 additional days, as you could see on this slide. The cell density was adjusted every 1 to 3 days to maintain CD4 plus T cell at a lower cell density and improve cell proliferation and viability. In terms of the data, as you could see on this graph, first of all, the cell viability remains stable for both conditions from 97 to 87% during the complete experiment, the 21 day. High cell proliferation rates were achieved for both DO level during the complete period. Taking into account the cell expansion in flask from D0 to D5 and the cell expansion in the bioreactor from D5 to D21, the average fold expansion of CD4 T cell at the end of the culture, or 21 day, is at 7% DO, 9.63 10 to 5 fold, and at 20% DO, 2.35 10 to 6 fold. Those results indicate a possible impact of the 20% dissolved oxygen on the culture of CD4 T cell compared to the culture performed under 70% of dissolved oxygen. The proliferation of T cell in an in vitro environment is also regulated by substrate and metabolite concentration. Metabolite monitoring during the whole culture process reveals comparable glucose consumption under both conditions. Cell density adjustment every 1 to 3 days by dilution allow maintaining glucose level without a narrow range, avoiding large concentration fluctuation that could definitely adversely affect cell metabolism. Additionally, lactate production was monitored at each dilution point. As expected, lactate level increased as the available glucose in the culture was consumed. As previously observed, the cell density adjustment reduced fluctuation of lactate level during the culture process. The fine control of those parameters is essential to maintain cells in a robust state and improve their proliferation. At the end, it is crucial to evaluate the functionality of the CD4 T cell. We did that by measuring the production of interleukin 4. This is the signature cytokine which is involved in several immunological responses. Bioculture was sampled at different time points, day 10, day 17, and day 20, and T cells were either left untreated or stimulated with PMA and ionomycin for 24 hours. The level of secreted interleukin 4 was measured in the supernatant using a human ELISA kit for interleukin 4. As we could see on those graphs, we were able to produce a functional T cell after 16 days inside the BioBlue single-use bioreactor under control of the DASBox mini bioreactor system. This present study illustrates the successful long-term expansion of CD4 plus T cell in a DASBox mini bioreactor system equipped with BioBlue 0.3 single-use bioreactor. The importance of controlling oxygen level inside the T cell culture have been emphasized in several studies, and we have seen a positive impact of a lower oxygen tension, 20% instead of 70%, without impacting the functionality of the T cell by interleukin 4 secretion. This study illustrates definitely the advantage of a controllable bioreactor-based culture system for the development of a scalable T cell production process. In conclusion, The DASBox mini bioreactor system offers a fully controllable culture system appropriate to generate high-quality, viable, and functional T-cell through the entire process, which will impact the quality of the data. We could summarize all those results in the following slide. The challenge of a culture, and more particularly the T-cell culture, are culture establishment and process optimization, requirement of a large cell number, switch from 2D to 3D, and having stable condition on small and large scale. Therefore, bioreactor is a vital tool to tackle those challenges by providing precise growth parameter control, scaling up opportunities for process development, reproducibility through standardization. I would like to say thank you for your attention. And if you want to know more about this work, you could use the QR code to have an access to the application note, which are available. Thank you very much.